And very important point, the study of fruit. What is saying? The study of fruits is called pomology. The study of fruits is called pomology. Okay. Three basic type of fruit are, so here first one is the true fruits are eucarps, and second is the false fruit or pseudocarp. False fruit means I told this type of the fruit develops from the thalamus. Now you can say true fruits are eucarps. They develop from the superior ovary, because ovary walls always forms the fruit. So it develops from the ovary containing functional seeds but not additional floral parts fused with them. Okay, means they have only the ovary and seeds, means carpel and ovary. Okay, because stigma style ovary, these are the part of carpel. Okay, so ovule and functional seed means ovule and ovary involved, and other accessory part means no floral appendages, no other receptacle or thalamus involved. So this is called the true fruits or eucarps. Example is the mango. What is the false fruit or pseudocarps? That develops from the inferior ovary. Inferior ovary means epigynous. Inferior ovary means epigynous condition. Okay, and together with this, additional floral parts are fused with the pericarp. Very important. Additional floral part means here thalamus. Thalamus gets fused together. Thalamus gets fused together. Thalamus or receptacle. Receptacle fused together. They form such fruit is known as the false fruit found in apple. Okay, false fruit is the example is apple. Parthenocarpic fruit, which we have already discussed. Now, not I'm going to again talk. Same classification they have given again. You can see that is simple fruit, simple fleshy. They are not talking about the dry indentation. Pyrus, malus, that is apple, pome, and pepo I told cucurbita, and then citrus, hesperidium, orange also, and then drupe, mango, which we have seen. Okay. Then here iterio of achene, that is aggregate, iterio of achene, iterio of berries, iterio of drupes, and iterio of follicles. Okay. And composite is the Syconus and Sorosis. So Syconus and Sorosis found in the jackfruit and Syconus found in the ficus. Okay. False fruit that I told already, apple. So apple, Pyrus malus, that is the false fruit. Okay. And Anacardium accidentalis, this is also false fruit. Okay. So this is the overall story about the fruit. Some part of the fruit, some part edible part they have also given. This is the lady finger and tomato. These are the simple fruit. Aggregate fruit, I told already. Aggregate means iterio of drupe, iterio of berries, iterio of achene, like that. Another very important thing, climacteric and non-climacteric fruit. I told already. During the ripening time, the respiration rate is become high. During the ripening time of the fruit, the respiration rate become high. So such type of fruit is known as the climacteric fruit. Now you can see. Fruits are defined as a fruits that enter climacteric phase. What is the climacteric phase? After harvest means when you have plucked fruit from the tree. Okay, they continue ripen. I told already. During the ripening process, the fruit emit ethylene along with the increased rate of respiration. Okay, already ethylene gas will arise when you have harvest the or plucked this fruit from flower or sorry, from tree. If you put anywhere, it will start to ripen. During the ripening time, they will start to secrete ethylene, which helps in the ripening. Along with ethylene, they also increase the rate of respiration. So this phenomenon is known as the climacteric fruit. So these fruit are harvested hard and green, but are fully mature and ripened near the consumption area. Fully ripened fruit are too delicate with a stand transportation over long distance. Okay, if we'll put for long time, it will be too delicate and it will be spoiled. Okay, so small dose of ethylene is used in induce ripening process under controlled condition, temperature and humidity. Climacteric fruits are mango, banana, papaya, guava, sapota, kiwi, fig, apple. Okay, so these are the climacteric fruit. I told what is the climacteric fruit. During the ripening time, when you have harvested from the plant or tree, so this fruit will start to ripen. Along with ethylene, they also increase the respiration rate. So this type of the fruit is known as the climacteric fruit. Example: mango, banana, papaya, guava, sapota, kiwi, fig, apple. Clear? Okay. Again, same thing. So I'm not going to talk about this non-climacteric, climacteric fruit. Okay. Now you can see what it have given. Non-climacteric. I told already, fruits do not show climacteric increase. Climacteric increase means their respiration rate is not going to increase. That is called non-climacteric. In the rate of respiration during ripening and do not respond to ethylene for ripening. And non-climacteric means if you are going to use of ethylene gas, it will not proper respond, means rapid ripening does not take place. So such fruit is called the non-climacteric fruit. Example is the citrus, grapes, pineapple, watermelon. Okay, so citrus, grapes, pineapple, you can see citrus, grapes, pineapple, and watermelon. So these are the example of non-climacteric fruit. Some part which is important, this is PYQS, lychee, the edible part of lychee. Point to remember, in lychee chinensis, the edible part of fleshy structure is known as the aril. Aril means this is the PYQS, edible part of lychee. Edible part of lychee chinensis. Lychee. Edible part of lychee is known as the aril. Edible part of lychee is known as the. Now it is okay. Let's start the difference between simple fruits and false fruits. Now you can see. False fruit means I told it is the means along with the ovary, some accessory floral parts they also involve, that is called false fruit. True fruits means only ovary involves. Ovary wall, that is ovary. Clear? So now you can read. False fruit arises from the floral parts other than ovary. Okay, means sometimes other than ovary or sometimes along with ovary. Clear? False fruit means they have arises from floral parts other than ovary. Clear? So this is the definition of false fruit. Okay. Now, true fruits arise as a result of fertilization, where the fertilized ovary wall becomes the fleshy fruit. Okay. It is saying true fruits means ovary wall involves in the formation of fruit. Means ovary involves in the formation of fruit. That is true fruit. And here, whether leaf means whether devoid of ovary or along with the ovary, some floral parts of the plants, this involves in the formation of fruit. This is called the false fruit. False fruit not involved. Fertilization never takes place. Fertilization does not involve. Okay. So in false fruit, fertilization does not involve, and here fertilization involves. Floral part such as thalamus, peduncle, and perianth parts of other than ovary, it involved. Okay. But here only the fertilized ovary involves. Clear? Example: apple, pear, jackfruit, pineapple, strawberry. Here mango, kiwi, fruit, watermelon, and cherry. So these are the true fruit. These are the true fruit. Now we have to seen already the difference between true fruit and false fruit. This is the structure of monocot seed. Means now we are going to talk about the seed. I told ovary wall forms the fruit and ovules responsible for formation of the seed. Now monocot seed, dicot seed means it is found in the angiospermic. Okay. So look it. Generally, monocotyledonous seed are endospermic, but some in orchids are non-endospermic. This is the NCERT line. NCERT. Endospermic means endospermous. They have the endosperm. Okay. This will be not consumed. And non-endospermic means totally consumed. Okay. So in case of the monocotyledonous seeds are endospermic, means they will consume their they retain their endosperm. Okay, but exception is the orchids. Orchids is the exception. Orchids is the exception. Clear? Orchids is the exception where membranous and generally fused with the fruit wall. Endospermous is bulky and stored food. In monocots, it is the bulky and stored the food. Now, the outer covering of endosperm separate the embryo by a protein layer called euron layer. Very important. In monocot seed, this is the important point. Monocot seed, outer covering of endosperm separates the embryo by a proteinaceous layer. Separates embryo by a proteinaceous layer. This proteinaceous layer is nothing but it is the alluron. Clear? This proteinaceous layer is known as the alluron. Is it clear? The embryo is small and situated in a group at one end of the endosperm. Embryo is small, it's situated at one end of the group, and it is the situated, and it will be the small also. It consists of one large shield shape. Very important. This is the PYQS. It consists of one large shield-shaped cotyledons, which is known as the scutellum. So scutellum is the characteristics of the monocot. Monocot seed, they have the large and shield-shaped cotyledon. The cotyledon should be shield-shaped. That is called scutellum. And short axis with the plumule and radicle. They have the plumule and radicle, but you can't say that plumule radicle replaced by the coleoptile and coleorhiza. Very short. Okay. The plumule and radicle are enclosed in sheaths, which is called the coleoptile and coleorhiza respectively. Suppose that this is the seed. In this seed, you can say they have the plumule. That is reason, but they have not given like radicle and plumule which found in the dicot. Okay. That is the reason we can think there is the plumule and here is the radicle, but not they have mentioned clearly plumule and radicle. This plumule and radicle region they covered by a special type of sheath, and the basic function of sheath provides the protection. So plumule and radicle enclosed in a sheath which is known as the coleoptile and coleorhiza. How you learn? Coleoptile, P came plumule, rhiza, radical, R came coleorhiza. So respectively. Okay. So it is the outer covering which protects to them, and coleorhiza which is found on the radicle which covered it is enclosed to the radicle, and coleoptile to enclose the plumule. Clear? Okay. Now again, this is the dicot structure of the seed. Dicotyledons store endosperm. Okay. But monocots store endosperm outside the cotyledon. This is the big difference. Dicot store endosperm where? Inside the cotyledons, but monocot store endosperm outside the cotyledonous. Okay. Plumule. Now this is the structure of monocot. This you can see. This is the structure of the monocot. They are going to store the endosperm outside the cotyledons. Okay. This is the seed coat pericarp, and this is the cotyledons. And the cotyledons of monocot cotyledons is known as the scutellum. Monocot cotyledons is known as the scutellum. Clear? Scutellum. Now you can see scutellum is there. Clear? And one outer proteinase, that means it is the enclosed, means it is the fused between pericarp, that is outermost layer, and alluron, that is innermost layer protein. Okay. So here you can say alluron layer, that is the made up of protein. Coleoptile, which is covered to the plumule, and shoot apical meristem SAM and root apical meristem, that is coleorhiza, they also covered. Okay. And plumule and radicle, these are very short. These are very short. Now you can see in this figure. In if you'll see in dicot, it will be big, but in monocot, it is the very short. Okay. So plumule and radicle are enclosed in sheath like coleoptile and coleorhiza respectively, which we have seen. Coleoptile and coleorhiza protect the embryo germination, but soon drop off. When the seed is going to germinate, it will be drop off. Vivipary is the characteristic feature which is found in the pneumatophore, which is the modification of the dicot root. Okay, pneumatophore is the modification of the dicot root. Clear? Pneumatophore. Now, sometimes seeds germinates within the fruit while attached to the plant. Such type of germination is called vivipary. So vivipary means when seed germinates and attached with plant, germinates and attached with the plant, this is called the vivipary. This is called vivipary. Vivipary. Clear? Vivipary. And vivipary is the characteristic feature of the pneumatophore. Pneumatophore means Sundarban delta. Pneumatophore. It is the characteristic feature. Okay, pneumatophore. So now you can say rhizophora, and Rhizophora mucronata and Heritaria. Toneratia avicennia, Rhizophora, and pneumatophore. So these are the uh, example of modified root, which we have seen already. That modification of root for the respiration purposes, which is found in the dicot. So this plant or this tree also shows the vivipary phenomenon. Vivipary means when the seed germinates, but seed gets attached with the plant. So this is called the vivipary. Found into the pneumatophore or Sundarban delta trees. Okay, mangrove vegetation. Now same things. Monocot again they have described. Look it. Yes. Just look at scutellum. I told scutellum it is the cotyledon. The cotyledon or monocot is known as the scutellum. Scutellum, that is a small shield-like structure, can refer to the equivalence of thin cotyledons in monocot, and especially members of the grass family. This is the PYQS scutellum found in the members of grass family. Very very important. Graminy. I told also yesterday LPG, Liliaceae palmaceae graminy. So these three are family considered as a monocot family. It is very thin and with high surface area and serve to absorb the nutrients from the endosperm during the germination. Okay. In the grass family, the cotyledon called scutellum. They have again the grass family where cotyledons is known as the scutellum. Situated towards the one side lateral of the embryonal axis. Now you can say towards the embryonal axis, only one side. At lower end of embryonal axis that has radical root cap enclosed by the undifferentiated sheath that is called coelurhiza. Now you can see this is the coelurhiza, which is the covering, which covered also root cap. Okay, outer protecting covering. The portion of the embryonal axis above the level of attachment of scutellum is epicotyl. Now you can see they have given the epicotyl. So this region is known as the epicotyl. Okay, epicotyl, and this epicotyl has shoot apex and few leaf primordia enclosed in a hollow foliar structure that is called coleoptile. So these epicotyl also covered with a special type of structure which is known as the coleoptile, and the basic function of coleoptile provide the protection. Again monocot seed. Now you can see endospermous and non-endospermous. This will be the clear your concept. Storage tissue, endosperm storage tissue, non-living, occupies the major portion in the seed. Alluron layer, I told it is the protein body which remains periphery of the endosperm. It helps in the absorption of water and release of enzyme which help in the germination, because alluron made up of the protein. Okay, alluron layer which is found in which is made up of the protein. Clear? Monocot seed is also called albuminous seed or endospermous seed. Okay, it is known as the albuminous or endospermous seed. Rice, maize, sorghum. Okay, albuminous means they have the storing substance as a albumin. Means during the growth of this seed, they will not complete consume this endosperm. Okay, rice, maize, sorghum. These are the example of the endospermous seed or endospermous seed or albuminous. Outermost covering of seed is known as the seed coat. We know inner integument tegmen, outer integument testa, both together called seed coat. The seed coat has two layers, outer testa and inner tegmen. They have again. Hilum is a scar on seed coat through which the developing seeds were attached to the fruit. Okay, where is the hilum is the scar. Suppose that this is the seed where the hilum is the scar. Okay, where is the hilum is the scar. So hilum is the scar on the seed coat through which developing seeds were attached. Okay, developing seeds were attached to the fruit. Above the hilum is a small pore. Here they have the small pore which is known as the micropyle. above the hilum, which is the small pore known as the micropyle. And you know micropyle is the side, micropyle is the pore by which water and oxygen going enters inside the seed. Okay, now you can see above the hilum is small pore, that is called micropyle. Within the seed coat embryo consisting of embryonal axis to cotyledons. The cotyledons often fleshy and food reserve food material. Food means it is full of the reserve food material. At the two ends of embryonal axis present at the radicle and the plumule. Okay, they have here clear cut radicle and plumule. Okay, now in some seeds such as castor, endosperm form as a result of double fertilization. It is food storing tissue. Okay, it is the exception is the castor. Like in monocot, orchid is exception. In monocot, orchid is exception, and here castor is the exception. Castor is the endospermous seed. This dicot is the non-endospermous, but castor is the exception, which is the endospermous seed. So now you can say some seeds such as castor, endosperm formed and result of the double fertilization. It is the food storing tissue. In plants such as bean, gram, pea, endosperm is not present mature seed. Such seed are called the non-endospermous. Why? Because non-endospermous or X-albuminous. In dicot, it is called the X-albuminous. Are you getting or not? Dicot seed, X-albuminous or non-endospermous. Why? Because during the germination of seed or maturation of seed, they consume their endosperm. But in case of the monocot, it will never consume. They reserve the food material into the outer side of the cotyledon. The cotyledon is the scutellum. So outer side of the cotyledon, they store the reserve food material. Okay, that is you can say simply non-endospermous. Okay, non-endospermous. It is found in the dicot, and monocot is the endospermous. Clear? Is it okay? Is it fine, Trishali?
Fine, sir.
Okay, now cotyledons, you can see again the structure which I have given plumule, and this is the dicot structure. Describe the structure of dicotyledonous seed. A seed coat made up of the two layer I told already segment, and they have the seed coat scar which is known as the hilum and developing to the fruit. Okay, this we have already discussed. So now this is the structure dicotyledons and plumule. Again, this is the figure which I have given in your NCERT. Now you can see that is the structure of dicot and monocot seed. Dicot and monocot seed. Okay, so look at here. This is the hilum. Hilum is the place by which they attach, and above the hilum, this is the micropyle by which oxygen and water is. Now, this monocot structure, you can see this is the endosperm, which is stored outside the cotyledons. Okay, this is the cotyledons, which endosperm stored outside the reserve, outside the cotyledons. Okay, and monocot, you have to learn very important point. I'm going to write monocot has endospermous seed. Endospermous means they will not consume. Endospermous or albuminous seed. Endospermous or albuminous seed. Clear? Monocot has endospermous seed or albuminous seed. Dicot has X-albuminous and non-endospermous. Why? Because they are going to utilize during the maturation. Okay, during the germination of the seed, they are going to utilize this. Okay, now exception in monocot, I told orchid you have to learn, and here castor. In dicot, castor is monocot seed. It represents the monocot seed. So here they have again scutellum. This is the scutellum where the reserve food material is stored. Coleoptile, plumule short, radical short, and coleorhiza, which is the outer protecting covering. Okay, so this is the structure of monocot seed. Okay, and now this is the protein covering inside the means outer is the pericarp. Okay, outer is the pericarp, and the pericarp, below the pericarp they have the alluron layer, protein covering. Okay, so this is the structure of monocot as well as dicot seed. The difference between monocotyledonous and dicotyledonous seed, you can see the basic concept here, it will be clear. Only single cotyledon is present with embryo, here two cotyledons, basic concept. Generally, cotyledon is thin and papery. Monocot cotyledons is thin and papery. Means why thin and papery? They are not going to store reserve. Means they are not going to store reserve food material too much quantity. That's why it is thin and papery. Here cotyledons are thick because cotyledons store this. Why? Sorry. Here just look at. Generally, cotyledons is thin and papery. Why? Because I told the reserve food material means endosperm. Endosperm is found outside of cotyledon. Endosperm is found outside of cotyledon. Outside of cotyledon. Due to this, generally cotyledon is thin and papery. Papery means paper-like. Papery means paper-like. So it will be thin. Why? Because they are not going to store reserve food material. Endosperm found outside the cotyledon. So in this way, it will be the papery. Cotyledons are thick because cotyledons store endosperm, that is the reserve food material. Generally, seeds are endospermic. Why? Because they store the reserve food material. They store endosperm. It is also known as albuminous seed. Albuminous seed. Exception is the orchids. Okay, exception is the orchids. Now, generally seed are not endospermic. Sometimes maybe endospermic. That is example is the castor. Castor means this is the exception, but most of the time non-endospermic. Cotyledon is called scutellum, which is found in the grasses, and here no, not called by the same. If seed plumule is covered by coleoptile and radicle covered by the coleorrhiza, seed is not found like this structure, generally absent. Okay, this structure generally absent. Now, again you can see plumule is lateral. Plumule is lateral in position and cotyledons in terminal position. So here plumule in lateral in position and cotyledons are in terminal position. What we have seen, if you will see the structure of dicot, monocot seed, they forms like that. Okay, so cotyledons here. So plumule is in lateral in position and cotyledons in terminal position. Plumule is terminal position and cotyledons are present. Just opposite. Here plumule in the lateral means sideways, and cotyledons in terminal position. But if you have learned one means that you have learned monocot. Monocot plumule in lateral position and cotyledons in terminal position. Just opposite means vice versa found in case of the dicot. Radical degenerate after sometimes adventitious roots forms at that place. I told they never bear the radical and plumule, but this type of the structure generally. Okay. Then radical is responsible for primary root, that we know very well. In some of the seeds, seed coats are cotyledons fused together, that is wheat. In wheat, you have to see seed coat fused together with the cotyledon, fused together with the cotyledon, It is found in only the wheat. Okay, and such type of seed is not found where the pericarp means ovary wall. That seed coat, testa and tegmen, they are not going to fuse. Okay, so this is the difference between monocot and dicot seed. This is the very important, and from here your concept should be clear. You have to learn endospermous and non-endospermous. This is the PYQS. Number of times they have asked. Okay, now you can see from here your concept will be clear. The difference between endospermous and non-endospermous seed. Endospermic seed. Endosperm is present in the mature seeds and serve as a food storage organ. Testa and sperm are two covering layers into the embryo. Means they have endospermous means monocot. Monocot they store mature seed and serve as a food storage organ. Okay, but in non-endospermous means dicotyledonous, cotyledons serve as a sole food storage organs, as in case of the pea, Pisum sativum. During embryonic development, you have to learn, this is the very important. During embryonic development, the cotyledons absorbs the food reserve material from the endosperm, because in the cotyledon, suppose that this is the two cotyledons where they have stored a reserve food material, endosperm. During the germination of seed, this endosperm is completely consumed. This endosperm is completely consumed. Clear? So that's why it is called the non-endospermous. It is called the non-endospermous. Non-endospermous always found into the dicot. Endospermous always found in the monocot. Exception is the archids in monocot. Exception is the castor in dicots. This I've given in your NCERT. Next, the endosperm is almost degraded in the mature seeds and the embryo is enclosed by the testa. Okay, endosperm is always degraded means completely consumed. I think from here this slide your concept clear, the difference between endospermous and non-endospermous. Is it okay? You can see again that is endospermic and non-endospermic seed. Endospermic means monocot. The plant embryo increase size only absorbs some of the endosperm core. Okay, they have reserved. Okay, they have reserved storage food material. Endospermous. Non-endospermous means completely consumed. The plant embryo increase in size, absorbing all the endosperm during the development. That is broad bean. Clear? So you can say they are going to consume all the storage reserve food material. Clear? So in this way, exception is here orchids. Orchids is monocots and the dicots is the castor. Castor is the exception of dicot. Okay, clear? So this is the overall difference between monocots and dicot seed. Okay, so thank